Cool. All right, we've got the recording going. And I say, Johnson, let's give it like a minute or so for people to join, and then maybe we'll even just quickly, introduce ourselves and let folks on the line introduce themselves as well.
Yeah, that works great. I've only heard from one person saying that they aren't coming. I believe everyone else is, correct, Margarita?
Oh, she messaged to say she can't hear us, but she's working on it.
I see that now. She's working on it. We're going to get there. It'll be good. But I believe there should be 11 of us on this call. 10 now, I think. So, 7, 8.
And Billiana is upside down.
I don't know. That's never happened to me before. Hello.
It's very amusing. I think we should all have one person upside down in every Zoom call just to keep us on our toes.
I've never seen, I don't know. I don't understand.
So are we all upside down for you or?
No, you guys are fine. It's
okay.
Just me.
It's just the webcam not liking you today. Did you have another call before this and it was flipped?
What's that?
Have you had any other calls this morning?
Yeah, I've had calls all day. I've never been upside down before.
Decided this is the one.
I'll see if I can flip it, but for now, here you go.
For now, it's actually it's pretty funny. Have you guys heard about Goat to Meeting, where you can pay to have a goat come up on your screen? And so yeah, I don't remember how much it is, but it's like maybe it's $40 or something like that to have like one of these meetings where there's an actual live goat that they'll put on the camera. So you can be kind of like our amusing. I'm not going to say goat, but amusement during the call.
That's awesome.
This sounds like an investment that we should put in, David, then.
That's right. Yeah.
A random meeting with goat.
So let's see. Kate, are you able to hear us now? I see your message saying that you could not.
I think she might have jumped off.
She jumped off and maybe she's going to jump back.
Yeah. Hi, everyone. It's Margarita. When I was joining and selected phone audio and didn't realize it, so I just hung up, and so maybe she did the same.
Cool.
That makes sense. Does that mean this 908 number is not you then, Margarita? That's someone just calling in?
Yeah.
Hi, this is Shah. Yeah, this is Shah. I'm on the 908 number.
Oh, fantastic. Thank you, sir. Yep. That way we can get a nice count in terms of videos and phone numbers because they show up separately.
Okay.
And then the last person just waiting for Kate Chang to join back. Kate, were you able to get audio?
Yeah, got it.
Oh, great.
Thank you.
Nice. Billiana, you've restored gravity. You're back.
I'm not on the moon anymore. We're great.
Nice. Well, is this everyone, Johnson, that you're expecting?
I believe we have one more that's not on here according to my count, but I feel like we should just get started. Cool. And if they don't show up, we'll just send them the link.
Yeah, we'll record it. Nice. So really quickly, just want to first off thank you guys all for videoing in and joining on to our Corona project. I'm David Fagenbaum. I run a research lab at UPenn that's focused on studying Castleman disease and related cytokine storm disorders. Obviously, our world has now been totally turned upside down by COVID-19, which also causes a cytokine storm. About 2 months ago, our lab decided that we wanted to shift a good bit of focus towards COVID-19. And one of the ways that we wanted to do that is around drug repurposing, the concept of taking a drug approved for one condition and using it for another condition. That's our best bet against COVID-19 because it's going to take a long time to develop new drugs against COVID-19. The first step in that process, in our opinion, of drug repurposing is trying to get a handle for all of the drugs that are currently being used. We knew there were a lot of drugs being used. I don't think we appreciated just how many there were until we started doing the work. But we went through a two-phase process. We're in the second phase right now. The first was to go through all of the published literature plus all of the pre-published literature on these medical archive websites, where we found over 2,500 COVID-19 papers. We systematically went through all 2,500 papers, pulled out data on 9,152 patients, and there were 115 drugs used in the first 9,152 patients, which was a lot higher than we expected. Now, there are a number of of compounds you'll see in that list that you won't recognize because they're not FDA approved. Some of them are supplements that are used almost exclusively in China, but we wanted to capture everything that's given to anyone. And so that number is actually up to about 150 now. So phase 1, we went through that first group of studies. Phase 2, and I should also say in phase 1, we spent excruciating detail trying to get a sense for which drugs were working and which drugs were not. So what day did the patient get discharged from the hospital? What day did their symptoms resolve? What day did their COVID-19 testing come back negative? Did they survive? We got a lot of detail on each patient, what were their comorbidities. And when we went to analyze the data, we found that even though we had all of the data on a lot of patients, there was so much missing data for so many patients. So a study might report 100 cases, and out of those 100 cases, it might say 50% are on one drug, 50% are on another drug, 20% of patients improved at this time, and 80% improved at that time. But you don't know if the 20% that improved were on one drug or another drug. There are basically a lot of holes. And so as a result, now that we're in phase 2, we've actually decided to be a little bit less ambitious as far as how much data we extract out, because we realize that even if we extract out everything from the case report, we're not actually going to be able to use a lot of it, just because there are so many gaps. So right now in phase 2, and what we are looking for you all's help with is to continue to go through the published literature. So every paper that comes out about COVID-19, and to look in that paper to see, is there a drug that's currently being used to try to treat COVID-19? And if so, enter it into the database, enter in the paper that it's coming from, and then just a few other data points. But it's really, we're moving from a situation where we wanted to get all of the data on every drug to now where we're saying, okay, our first goal is just what's being used. We need to know, if you're fighting a war, you want to know what weapons your front lines have used, and you want to get a sense for which ones are working and which ones aren't. So the goal of our project, the Corona Project, is to really get a good grasp for what are all the weapons that doctors are using around the world, get them in one place, how frequently are they being used, and then some really basic information on whether they're working or not, but not the kind of detail from the first pass. Johnson, anything else that you would add to that high level, but also very detailed summary I just gave?
No, I think that's really a great overview. We'll be looking at both drugs that were given to patients, as well as we'll talk a little bit later, drugs that are done in vitro studies where we think that it could be used in the future. So we have a little database of that too. So there'll be a little bit of that distinction that we'll talk about later too.
Yeah, that's great. Thanks, Johnson, for adding that important point on. Maybe before we dive into the training, I don't know if you guys want to quickly go around and say who you are and what you guys do. It would be helpful for us just to know you guys' backgrounds. And so I guess I could call on people. Zoom can get really disorderly, so I'll just call on people if that's all right. Maybe Maggie first.
Hi, I'm Maggie Riley. I'm calling in from Charleston. I used to work at Amicus. I was an intern for a couple of summers up there, but now I'm a medical student at MUSC down here. So
we're thrilled to have you. We've had a number of medical students, almost half of our team, and it's been great to have. It's been awesome. So glad to have you on the team.
Great, thanks.
Helen.
Hi, I'm Helen. I'm at Amicus. I work in the structural biology group, specifically on protein engineering.
Nice. And are you in the Philly office? Well, I guess you're not in either office, but are you typically in the Philly office or the New Jersey office?
I'm in the Philly office, so I'm actually in the Philly office.
Oh,
so,
wow.
Had some lab work today. So
Cool.
attended the conference, the Gene and Cell Therapy conference this morning, and I'm doing some lab work this afternoon.
Cool. Nice. It's great to have you on the team, Helen. Margarita?
Hi, everyone. Yeah, so I'm actually now a patent lawyer, but in the past I was a biologist. So I have a PhD from Rockefeller University where I did structural biology. I did a postdoc before I eventually just left the lab and went to law school, but I really enjoy the science, and I think this is particularly relevant and interesting, so I'm really excited to be able to participate.
Thanks, Margarita. Aura?
Cool.
Hi, my name is Ora Enake. I'm with Amicus, and I'm particularly in the discovery biology department. I have joined Amicus in January, and previously to that, I actually worked for Thomas Jefferson University in the lab of Dr. Al-Namri, biochemistry and molecular biology.
Wow. Wow. I will say what I'm hearing so far is that you guys are incredibly qualified, and I'm super impressed. You probably will not need all of the degrees that you guys have for this particular project, but it's awesome to have you on the team.
Thank you.
Yeah, Billiana.
Hi, I'm also at Amicus. I'm in medical affairs now, but my background is in neuroscience. My PhD was in developmental and molecular mechanisms of neurodevelopment.
Wow. Nice. Glad to have you on the team. Heather?
Thank you.
Hi, I'm at Amicus. I did some work previously at New Jersey Medical School in their oncology hematology department. And I worked in oncology CRO for a few years, and now I work in clinical operations on the Pompe disease.
Nice. Oh, that's awesome. Thanks so much, Heather. Kate?
Yes, hi. I work at Amicus, and I have a background in biochemistry, and currently I'm working in vitro pharmacology, and I also have a background in bioanalytical science.
Awesome. Thanks so much, Kate. Jason?
Hi, everyone. I'm Jason Wisk. I work at Amicus on the medical team with Ileana. I'm a physician, and I made it partway through my training in radiology at Duke, but left it for this role.
Cool.
So far, so good.
Nice. Glad to have you on the team. And are you still in North Carolina, or are you up this way?
So I'm quarantining in North Carolina. I've been here for 2 months. I'm normally based out in Arizona and California.
Oh, wow. Wow.
Yeah.
Cool. Aaron?
Hey, guys. Aaron Corona, employee at Amicus in the New Jersey office. I'm a project manager in our regulatory affairs department, and I lead some of the volunteer efforts for the company. So when Margareta presented this to us, it was very exciting because a lot of employees are looking for ways they can volunteer and ways they can volunteer to give back to the COVID efforts and ways they can volunteer at home. So this is an awesome opportunity. So looking forward to hearing more about it.
Yeah, that's great. And then there's 2 more folks on. I don't see their names. Feel free to jump in.
Hi, this is Shah. I work with Margarita in the legal department at Amicus. I'm a patent attorney by training, 25 years with Big Pharma and now with Amicus.
Wow.
Most of the work I do for Amicus, though, is transactional work. My background is in pharmacology and medicinal chemistry.
Cool. Wow, that's awesome. And then
hi, I'm Philip Rubichinski, and I'm at Amicus. My graduate work is in organic chemistry. I work at Amicus now, helping the researchers to collect their information and then to archive it.
Cool. Well, that sounds very relevant. We're trying to collect and archive a lot of information right now. That's great. So maybe I'll kick it over to you, Johnson. Of course, feel free to introduce yourself as well. And John, so maybe I'll give a quick intro for Johnson, and then he can go further. Johnson is the coordinator for the center that I run at Penn, focused on cytokine storms, and has done a really great job coordinating this project in particular. So Johnson, take it away.
Thank you very much. Like David said, I'm a graduate student here at Penn, and I also help coordinate the Castle and the work that we do and liaise between our research and our clinical sides for Castleman disease. So I'm going to share my screen now. So you guys should be able to see this Google Drive right now. You guys all, once you send me an email that you'd like me to share access to, I'll send this to you. A couple of you have already, which is great. Thank you very much. This is what you'll see when you first get into the folder. There's not a lot here, which is good. We try to make it as sparse as possible so that there's only a certain number of things you'll need to access. The first thing that we'll talk about really quickly is this protocol sheet, which is also in the email that I sent you all. So you guys should have all this information there, but it's also here in case you want to pull it up side by side when you're reading through things. Rather than read through this protocol, we're just going to implement it. Hopefully you guys were able to take a look at the protocol and take a look at the paper that was sent out last night. If you didn't get a chance, that's completely fine, because we'll do it together. The next thing I wanted to bring up is, oops, wrong tab, is our spreadsheet. So this is the spreadsheet that we're going to be extracting all of our data from. This first sheet right here is our summary of drugs publication sheet, and that's just going to be where we're taking the information and actually extracting the information about the different drugs that are being used. So as you see, there's the treatment name here, there's frequency. This frequency is actually a, this up-to-date frequency is a formula, so you don't actually have to update it yourself. It'll update itself, which is nice. And then we'll have all of our PMIDs up here on the top, and we'll go into detail on how to use it really soon. Next tab that I want to bring your attention to is this listing of studies to do, and this is where we're going to list all the studies that have been published in PubMed and how we're going to go about them. I picked the one study that we're going to be talking about today, which is this yellow one, so that it just comes to your eye. When I picked the study, there was nothing taken around it, and so as you can see, in a day, basically, people have come through and read through most of these articles coming before and after, which is really great. And this next tab is our summary tab. And this tab is where we're going to, once again, extract that information, but it'll give us a little more detail in terms of how many patients are on each drug, and then whether or not there's an assessment. We've partnered with CureID, which is a partnership that David could talk a little bit more about later if need be. But they asked us to really try to see whether or not these drugs are working. Can we say from the case report or whatever paper that we've seen if there's an improvement, if there's a deterioration, or if it's really just summary level data. And then the last tab to talk about is this in vitro studies, which we'll talk about at the very end after we go through our case report. So the first thing that we're going to do is really just simulate what it'll be like picking a paper and going through the whole thing. So the first thing that you'll do is in this first column, column A, you'll put your initials. This just allows us to make sure that multiple people aren't working on the same paper. And so put your initials down. Some people like putting down one initial, doing the entire paper, and then moving to the next one. Some people, as you see, like taking a huge chunk of them and then moving through line by line. Completely up to you, whichever you'd like to do.
Oh,
this first column.
Oh,
yes.
Sorry to interrupt. Maybe just scroll down and show, so like what they would do. So they would find an empty place, because there's like an empty spot there. So this is just like our listing from data that we extracted a couple weeks ago. So we pretty soon need to update this. We're closing in on finishing up all of the papers from the last couple weeks. But yeah, you would find an open spot like here, and then throw your initials in. So I just wanted to make sure it was clear that that you find one of these open rows and throw your initials in.
Yes, sir. Let me find my yellow again. So if we were to put, I just put my initials there, that way it's just taken for this training. The first one is reviewed. So this is really just a track for you, just to see whether or not you've read it or not. Anytime a paper has been read by something, you'll put yes here for reviewed. And so what you'll do, easiest way, take this PubMed ID number, bring it into PubMed, which I'm sure you all know how to do. I'm going to go through a little more detail just so that we can send this video out for other people who might not have the training that you all have. So bear with me here. So if you search that PubMed ID, you'll come to this. It'll give you, most cases, a nice abstract and then a full text link here. You're going to want to click this full text link so that you can get it. A nice thing for individuals who might not have as much experience with PubMed is if you click this cite button, it'll just give you the nice citation. So you can just pull it and put it onto the spreadsheet, which we'll do in a little bit. So this is the paper that was sent out yesterday night, high resolution chest CT features and clinical characteristics of patients affected with COVID-19 in Jiangsu, China. I apologize if I said that wrong. First thing you're going to want to do is find this download PDF button. Every single journal has it somewhere different, and so sometimes it's a little bit of a search to find that PDF. We're going to move us over here and bring this up here. That way we can have both simultaneously. If you were able to read the paper, that's great. If you weren't, high level sort of idea. This paper was really looking to see with a multi-center retrospective study whether or not you can use a chest CT at the very beginning at intake to see whether or not patients have COVID. And so you'll read through the paper. At this stage, we're not going to be seeing a lot of case studies, so it won't be immediately evident whether or not there's drug repurposing in the study. We're considering any drug that's being given to a patient as drug repurposing. as there's nothing that's approved for COVID yet. So if we read through this paper, talks about what they're doing, why they're doing it. I've read through the paper, so I'm just gonna scroll to where we need to go. But you'll read through the paper, try to figure out what's there. It's nice that you get a little bit of background on the paper. What you're gonna wanna do is you're gonna wanna highlight directly onto the PDF anything that you're taking out. So actually we'll go to here. So it says that it's a multi-center study including 234 patients from 13 hospitals during 17 days. So we're gonna highlight this number so that we know there's 234 patients that are in this as a whole. And so we'll go to our Corona sheet here. We'll go to here we'll put yes. We'll put that no, it's not been previously extracted. This column we were having you guys fill out, but a medical student actually wrote a script that went through all of them, so you guys don't have to worry about that. We'll say yes, that it's being extracted. And then we'll move on to this summary of extracted studies pub. The first one, I just went to the top of every sheet, but the first 156 entries here were from our publication. And so they're gonna be formatted just a little bit differently than ours is. So we'll go to the very end here. As you can see, there's a lot of entries here. We wanna make sure that every single drug gets its own line. Well, I guess there's a couple exceptions, leponavir and ritonavir, usually given as one drug together. And so those can be on one line. Amoxicillin, and this one is also one line. So you'll see a couple of here that are just one line. All right, so I neglected to say that the first thing we should do is fill this out. So we're gonna put our citation down, just last name and the date. So it'll be H 2020. We know that from here, multi-center study, so we'll say a series. The author in here is China. And then we know that the number of COVID-19 patients that are in this study is 234. Clearly, I cannot spell. 234. And then, so 234, we'll scroll down to where I know this information is. But right here at the end of this paragraph, it talks about drugs that were used for these patients. And so as the therapy schedules, patients received antiviral therapy, which is either oral, intravenous, antiviral drugs, and inhalation of interferon. So we'll put this down here, unspecified antivirals. We'll put down that they also got interferon. If you'd like, you can copy and paste. I tend to just type it out, but copy and pasting works well to make sure that you don't have any spelling mistakes. You can see that each patient was given it so that we know that all 234 patients got each of these drugs. Read further, antibiotics were administered in 118 patients to prevent or treat concomitant bacterial infection. So we have unspecified antibiotics. That was 118. Then we have our methylprednisolone. And that was given to 34 patients. And then we have our gamma globulin, which was given to 34 patients. And the last thing here is non-invasive ventilator, and we're not collecting ventilation, so you don't need to input that at all, even though it's given to only severe and critically severe patients. So we know that number of patients given specific treatment has been entered, and then we're on this cure ID. If it's immediately available to you as to whether or not there's improvement, deterioration, something like that, feel free to put this information in. This paper, as we know, is not patient level data. So we can just input that and put down for all 4, or I guess 5 of these options. And then you'll put your full citation, which can be found at this lovely site link here. So we'll just copy all of this and throw it in here. And so that right there is how we're gonna fill out this one form here. Do you have any questions on summary of extracted studies before I move on? I think it's pretty self-explanatory, which is great. So the next thing you're gonna do is you know that you have these drugs that were given to these patients. So you're gonna go to the summary of drugs pub sheet and input that same data over. And so you're gonna go all the way to the end here, wherever that is. Here we go. You're gonna copy this PubMed ID that you searched for the paper, and you'll copy it here so that we know that all the treatments that are in this row DM were given in this one paper. And you'll do the same thing over again in terms of all of these drugs. So if there are any unspecified drugs, that's going to be in this non-specific category. So we know unspecified antiviral, DM, there were 234. In terms of interferon, you can search here so that interferon, you can search so that it comes up a little bit faster. The downside of searching is that you then have to scroll all the way across again. So depending on you, whichever one you'd like to do. This one's also going to be 234. We go down to our next one, which is antibiotics. So that's unspecified antimicrobial. That'll be 118. We have methylpred. I think it's a little bit easier to search and then scroll. If you decide that you'd like to scroll up and down and search it yourself, that's completely up to you. 34, and then we have gamma globulin, mainly because I can't read fast enough to find this information. So 34, and then the last one is ventilator again, which we don't need. And so once you put all this information here for DM, it'll auto-populate here in terms of your up-to-date frequency, so you don't have to do anything like that. This paper didn't have any new drugs that weren't being used. If you were to find a drug that was not used in a previous paper, you'll come all the way down here to the bottom, and you'll add the drug name, and then you'll add, just like before, your PubMed ID on the far right, you'll put in that one, and then do a drag down so that it'll populate the formula downwards, and then you don't have to recode anything. This ATC code will input at the end, so you don't have to search that up either.
Oh, Johnson, just one thing I'd mention. So
Yes,
from the first few, I guess maybe the first couple thousand that we've gone through in this phase 2, there have been about 30 drugs added that were not part of the first the first 9,000 patients. There were 115 drugs. The last 3,000, there have been about 30 new drugs. Sometimes you find a drug that's got a name that looks like it's new, but actually just do a quick Google search before you add a new row, because sometimes it's actually probably half the time it was actually already above. It's not the end of the world. We can always double check your work, but it's just worth doing a quick Google search. If you think it's new, just make sure there isn't another name for it that's not already listed above. And by the way, when we enter in names, you'll notice we enter a drug like rituximab. So that's the generic name. We don't put Rituxan. We put bortezomib, not Velcade. And so you're always putting in the generic name. But even sometimes there are different generic names depending on what part of the world you're in. So it's worth doing a quick Google search to make sure that it's not already in there somewhere.
Thank you, sir. So then once you've read through the paper and you've looked for all the relevant information, this paper does not have any more relevant information, you're going to save the paper onto your computer, wherever you are, and then you'll go back to this COVID-19, Google Drive, and you'll go to annotated paper PDFs, phase 2, and then you'll upload the paper into this folder. This is just so that we have a check as to the papers that are being read and extracted, and that if we need to go back in the future, we have sort of this highlight that tells us at a quick glance where the information that you pulled from and where it is. This also helps in case we do an audit later down the road. Do we have any questions about what we just did in terms of just extracting this information before we move on to our last sheet, which is in vitro studies?
Oh, Johnson, sorry, real quick, Johnson, will you head back to, I think, did we just lose Johnson?
No, I'm here.
Maybe you stopped sharing screen. Oh, cool. Will you just go back to that first sheet and around there's a column around response, right? And sorry, we're not seeing your screen anymore.
Oh, it says we are screen sharing. Let me stop sharing and reshare then. Can you see it now?
Oh, yes. Thank you. Sorry about that. Okay, cool. So yes.
Column G.
Good. So you went ahead and put in not patient level data, which is good. So I just want to clarify that, and maybe I missed it. Maybe you went into it in detail, Johnson. But if this paper said that for the 234 people who got interferon, that all of them improved, then you could put I would then put 234 improved to be clear that, all 234 improved. Just to give a little background, there's an app that the FDA developed called Cure ID, which is just trying to categorize every drug that's being given to COVID-19 patients as either improving, deteriorating, or undetermined. And so we are trying to help them out by just trying to categorize every drug into one of those three categories. And I know that's really hard to do for a lot of reasons, but you're just basically going to try your best to figure out which of these three categories does it fall into. So even if it's summary, so like not patient level data, but it's summary, but it actually says that, all 234 improved, you can put 234 improved. If they say that 10 improved and you feel like the other 224 deteriorated, you could try to put that in as well. This is just our attempt to help the FDA folks out, but it's not very easy to do. So that's why you'll notice that it's not filled out in a lot of cases. Johnson, maybe you went into that depth already and I missed it. Was that a good way to describe it?
Yeah, of course. And then just to add a little bit more nuance, there is this undetermined option. Feel free to use it. We've noticed that some people don't want to put it down. They would rather leave it blank than say something like that. There's a reason it's here. So please try to keep this column G filled out. Every line that has a drug should have some sort of response attached to it. If you are unsure about the response, you'll all be added to a Slack group, and that's a great place for you guys to post the paper and say, I'm a little confused, what do people think? And we can come to a consensus that way. That's also an option for you, but please we're trying to avoid blanks as much as possible because they're a little bit ambiguous, and that then causes one of us to have to go back and try to figure out why that blank is there and how we can best fill it in. I heard another voice earlier.
Yes, I did have a question.
Yes, ma'am.
Instructions, you mentioned something about prophylaxis.
Yes.
Can you elaborate on that or give some examples?
Yeah, so we're not inputting any drug that was given prophylactically. So if we're saying that there is a clinical trial that says they're giving, let's say, hydroxychloroquine to all the physicians that are working there, we won't be extracting that information because it's not used to treat COVID. It's used to try to prevent COVID or try to mitigate symptoms and things like that. And so that's a little bit outside of the scope of what we're trying to do here, where we're looking at just what drugs were effectively used. And that also moves into the fact that we won't be using any drugs that are, well, on this sheet, we won't be extracting any drugs that are being proposed as a good option in the future.
And one thing I'd add, just to make it, at least it helps me to think about it, is that this is a database of drugs given to people with COVID-19. So if there's a trial of hydroxychloroquine to prevent COVID-19 and 10 people get COVID-19, you can enter those 10 people in and say that they got hydroxychloroquine because they're people with COVID-19. That's fine. So the distinction is not so much whether you're using it preventatively or not. The distinction is did they ever have COVID-19 or not? If they didn't have COVID-19, they don't go in the database. If they did have COVID-19, then they go in.
Okay, thank you.
Perfect. Any other questions before I move on? No? Fantastic. So this last one that I like to bring up is our in vitro studies extracted sheet. This sheet is very similar, as you can see, to the summary of extracted publications sheet, with the exception that it's drugs that are being proposed for use later. And so you'll do basically the same thing, your citation, your country of author, what the proposed treatment is, and then the difference then is a short rationale for the proposed treatment. So feel free to copy paste exactly from the paper saying, like, we think that this drug or these drugs will work effectively against COVID-19 because X, Y, and Z. We don't have this for all the papers. This was something that came up about a week into we starting phase 2 where there was interest in this. So if you encounter papers like that, feel free to enter this information in here as well. Fantastic. So I'm going to stop sharing my screen now. That way we can see all of your faces. Or at least I can see all your faces, so I can't see anything. And see if there's any last questions that you guys have in terms of entering information in, trying to find this information, things like that, or anything that you'd like to ask just to close us out.
Johnson, maybe a question for you. Can you tell us the proportion? So we've gone through over 5,000 papers now. It might be closer to 6,000 by now. So pretty soon we'll be uploading the next batch of the last 2 or 3 weeks worth of papers, because that's about how behind we are. We're about 2 to 3 weeks behind, which is why, in particular, why we're really happy that you guys are going to help add some horsepower, because I think that in a crisis like we're facing with COVID-19, up-to-date information is really important. And the closer that we can get to real time, the better. In your opinion, Johnson, out of the cases, what proportion of papers that you look through, what proportion of them actually have a drug that's been given to a COVID-19 patient versus, let's say, a commentary or a in vitro study? What's the proportion of papers that have drug repurposing you'll extract out?
Yeah, so I think phase 1 and phase 2 very different in terms of the types of papers that are coming out right now. Phase 1, we had a lot more case reports that were very easy to spot as like, this is going to be drug repurposing. The one that I chose, high resolution CT features, might not seem as something where you have drug repurposing, which is one reason why I chose it, being that it's important to at least give these papers a skim and to just see, like, even though it might not seem like it, there could be information. Like the information we got was from 2 sentences in the middle of the paper.
Yep.
And so I think at this point, it might be 1 in every 15, 20, where you have drug repurposing. And so you might run through a list of them where you don't see any drug repurposing, and that might be fine. If you have another nuance to bring up is that when you look at the title, if it's in brackets, that means PubMed has designated that it's not in English, or the original article is not in English. So feel free to Google Translate if you want. If you have the capacity to read that language and you'd like to read them, feel free. We do have, as we know, COVID-19 really started in Wuhan, China. That's where most of these case reports in the beginning are from. We do have an individual who speaks Chinese fluently and says that he is more than happy to read through them. So if you find them in Chinese and you'd like to pass them along, let me know, and I'll pass them along to him. If not, feel free to Google Translate or just notate that it's in another language and you can't read it, and someone else can pick it up then.
Yeah, so thanks so much, Johnson. So I guess an important takeaway is that there are going to be a lot of papers that you skim through that there's going to be no extraction done at all. And we don't do any sort of like follow up to see, like, did that person make the right decision or the wrong decision? So this is, your shot to figure out, is there a drug being repurposed? I think, in my opinion, and from my experience, the abstract should be able to give you a pretty good sense. In the abstract, if it doesn't mention any patients being treated, it's going to be pretty unlikely that the paper is going to have anything about a patient getting a treatment. At the end of the day, you're just looking, are there patients in this paper, and are there treatments given to those patients? So use the abstract to the best of your ability. Maybe skim a little bit if you don't see something in the abstract, but be happy, or be okay with the fact that there are going to be a lot where you're not going to find treatment. And actually, one thing you can always do, we need to get through every paper, but some people will read the titles of the paper to see if they can enrich for the ones that sound more likely like there's going to be a drug, and that's totally fine. We need to get through all the papers, but if you're more interested in going through the ones where there's something to extract, then that's great. Someone might actually be more interested in going through the ones where there isn't something to extract, and that's really important work to go through and say, oh, I can confirm that this doesn't have any drugs given to patients. I can put an N in the list. That's also really valuable. So it's really you guys' call.
Perfect. Do you have anything else that you want to say, David? Or
yeah,
else?
The only other thing, and I'm sure we're running up on time. I don't know if you guys have passed 1:30, but the only other thing I'd add is I think I kind of glazed over at the beginning why this is so important. When we have an emerging disease where there are no FDA-approved drugs, it's really important that the existing drugs, the over 1,000 FDA-approved drugs, are considered as potential drugs for this new condition. And as you can see, a lot of doctors are trying a lot of things. And so this is our best effort to say, okay, a lot of things are being tried. We want to try to pull it into one place, because no doctor can be expected to read through 5,000 papers. But that doctor can look through our simple list and say, oh, wow, these are all the drugs that have been tried. I could consider using one of them. that's probably a lower on the priority goal. Probably the number one priority for us is let's come up with a listing of drugs so that researchers and companies can say, oh, wow, our drug is being used a lot, or, oh, wow, this drug is being used a lot. Maybe we should progress it to a randomized controlled trial, or, oh, it's been used 3 times. Let's look at the original data and see how well it worked in those 3 times, and then think about moving forward to a trial. So I think this is number one, to say, let's see what's being given so we can figure out what trial should be done. And then number 2 is, let's just inform the world, doctors, physicians, doctors, researchers, healthcare organizations, about what's been given, which could potentially inform treatment decisions. Any questions for Johnson or I, or any comments? Johnson alluded to the fact that we had the first paper, or the paper from our first analysis was recently accepted for publication. So that'll be coming out in the next week or so, which will hopefully lead to there being additional attention for the database. We're spending a lot of time on it. We want to make sure that this is useful to people.
That would be really interesting to read. Would you be able to circulate it, or wait until it's published?
That's a great idea. No, we'd be delighted to share. I think that, we can just share the PDF, and then Johnson, you should be able to find it pretty easily on the Z Drive, the one we submitted, and or the final submission. So yeah, absolutely.
Great.
Yes, sir. I can circulate that.
Make sure that the supplementary appendix goes with it, because we have a big supplement, and the supplement's got a lot of good stuff in it around drugs being used.
Great.
Yes, sir.
You alluded to timelines, and we were some of the new volunteers asked about time commitments, and I think we have varying amounts of time to spend on this. But I sold it as you can do 1 paper, or 300, whatever time you have. And I think we all don't always know how much time we will have. But I wanted to know if there's a basic expectation, or what if there's a deadline that you're trying to meet, what numbers should we shoot for, and things like that.
Great question. So we have no minimum. We are just delighted and thankful for anyone who wants to put the time into this. So no minimum. If you do 1 paper, then that's okay. We thank you for your paper. We hope that you will do more than 1 paper, because we do have a big backlog. Johnson, you and I haven't looked to see how big the backlog is, but it's unbelievable how many publications are coming out. Thankfully, there's so many publications, but I bet we're probably 2,000 or 3,000 papers behind. And so, we're hoping to catch up, but I don't want any one person to feel like it's their responsibility to catch us up. But so I'm not really giving you a good answer, but maybe Johnson and I will look at how behind we are, like how many thousands behind we are, and see if we can set some sort of like an internal goal for all of us. And it wouldn't be on a per person basis, but just on average, this is about how many papers we'd like for you to get through.
Sure, that'd be great.
Yeah. Johnson, let's do that.
Yeah, I'll make a note. Perfect. Any other questions?
I just want to go back to what you said about outcomes too, because I wasn't 100% sure that I think, so I haven't looked at the papers yet, so maybe it will become more clear, but, sometimes there's no outcome. So what's it like, what do you do with that in the end? And how do you make sure, if there's certain numbers of patients had this outcome, and maybe they received 2 different drugs, and, I can see how it could get confusing. So I'm wondering if you can just talk a little bit more about that.
Yeah, we like I was saying with our phase 1, we extracted everything, and we were so ambitious, and we wanted to use everything we extracted. And just as you said, we really were not able to, because it might say that 80% of the people in the study survived, and 15 different drugs were used, but you don't know which of the 80% survived on a per drug basis. You just know that out of the group, 80% survived. And so it becomes really, really complicated. So for us, we just added that column in for that FDA app, and that app, Cure ID, they're just trying to bucket every drug that's given to patients into one of those 3 categories. And as Johnson said, there are going to be a number where you're just going to really have a hard time bucketing a drug into one category. And in fact, I think it's unlikely that every patient on that drug will easily fall into one category. So you might have to put like 10 out of the 15 patients resolved, 5 out of the 15 did not. So it's kind of try your best. And I've warned the FDA folks, I'm like, this is the best we can do. It's not easy. It's not clear in these papers. So they know that the data isn't great on the kind of the response. But they did say that when they go to move it into their database, that they'll try to do some sort of auditing process. So just kind of try your best. Johnson, is that how you would say it?
Yeah, definitely. There are papers that, like the one we read, where it's not going to tell you much. It's just going to say they got a drug and we moved on. And there are also other papers that are going to say they got this drug and they recovered, or they got this drug and symptoms resolved. And so you do get a little bit of both. I'd be looking for that correlation and trying to make the author tell you what they had. If they don't tell you, use that undetermined option. That's a great option to use, and feel free to use it rather than try to really sit down and spend like 20 minutes trying to wrestle out what you feel like is the best option there. If the data's there, put it in. That's what we need. But don't try to spend days looking at it.
Okay.
And you probably saw that from what we're extracting out, it's much less complicated than it was in phase one. Phase one, we were literally pulling out so many different data points. We were inferring so many different things. This is a much more simple extraction. Hopefully it'll go a little bit faster, but I know that for some of you guys hearing your backgrounds, I'm sure you probably would have been like really interested to do some of the really complex sort of conceptualizations around, is it this drug or that drug, and what are the proportions? And so I guess I'm sorry that we can't give you that level of complexity, but I'm also you're welcome, because it was a real headache for us in phase one. So this is a much more straightforward process. And hopefully you guys will also find it rewarding, knowing that we're providing this service. Cool. Without any questions, Johnson is always available. I'm available. We've got the Slack channel, so feel free to put messages in there if you have any questions for other data extractors.
Yeah, and before we leave, please send me an email that you think would work well. That way I can share this information out. I believe 2 or 3 of you did. But before that, I don't know where to share it to, and so you don't have access to anything. So send me that email, and then I'll send out the paper later on today.
And the other thing I would say is feel free to share with your coworkers or friends or family, other people that you think would be interested in joining on. This is recorded, so we'll be able to share the training with others. But as you can tell, it's going to take horsepower for us to catch up to real time. So anyone that you think would be interested in rolling up their sleeves and trying to help out in the Corona fight, that would be awesome.
Sure. Yeah. Thank you for having us be a part of it. It's really exciting.
Thanks for organizing this.
Thank you, and thank you to everyone else.
Thank you.
Great. Thank you.
Have a great day, y'all.
Okay, bye-bye.
And Johnson, so you're sticking around. I will stop recording.